Hello everyone. So far we have discussed about lipid bilayer in detail, about its structure, fluidity and asymmetry, etc. But we also know that a cell membrane is not made of lipids alone. In fact, proteins are equally important because they carry out most of the membrane's dynamic functions. So in today's lecture, we will focus on the classification of membrane proteins and how they are associated with the lipid bilayer so that it will help you to understand their function. So let's begin. These membrane proteins, they are majorly classified into two proteins. One is the integral protein and second is the peripheral proteins. Now these integral proteins, these are the ones which are embedded inside the membrane. Like here you can clearly see that this is the outer leaflet of the lipid bilayer which is facing towards the extracellular side, and this is the inner leaflet of the lipid bilayer which is facing towards the intracellular or the cytosolic side of the cell. And this structure which is spanning through the lipid bilayer is the integral protein. And since this structure is tightly bound within the membrane, it is not easy to extract them. For extracting these proteins, we need to disrupt or break this lipid bilayer. And for doing so, we need detergents like STS or Triton X100, while there are other techniques which can be also utilized to extract these proteins, which we will be studying in our upcoming lectures. Now, second is the peripheral proteins. Now, as the name suggests, peripheral proteins means these proteins are found at the periphery of the cell, whether being the extracellular side or being the intracellular side or the cytosolic side. Like here, you can clearly see that this is the peripheral protein which is found outside the cell, and this is the peripheral protein which is facing towards the cytosolic side of the cell. And since these peripheral proteins, as you can clearly see that these are not embedded inside the lipid bilayer, instead they are loosely attached with integral protein or with the polar heads of the lipid molecules, basically via non-covalent interactions like hydrogen bonding or electrostatic interactions. And since these peripheral proteins, they are loosely attached, so they can be easily extracted by using salts or by changing the pH. Now, next, if we talk about these integral proteins, these proteins are further sub-classified into transmembrane proteins, lipid-anchored proteins, and monotopic proteins. So, let us now study about these different integral proteins in detail. Now, these transmembrane proteins, these are the ones which spans the entire lipid bilayer. So, they have both the hydrophobic as well as the hydrophilic regions, where the hydrophobic regions, they interact with the hydrophobic core of the lipid bilayer, while the hydrophilic regions, they are exposed on either side of the membrane. Now, since these proteins have both hydrophobic and the hydrophilic regions, so they are considered as amphipathic in nature. Next, these transmembrane proteins, these are further subclassified into single pass and multi-pass transmembrane domains. When in case of single pass, the polypeptide chain spans only once across the lipid bilayer. While in case of multi-pass, the polypeptide chain, it spans multiple times across this lipid bilayer. Like here you can see that here the polypeptide chain is spanning 3 times across the lipid bilayer, right? So here the polypeptide chain, it can be either alpha helical or it can be either beta sheets, where the beta sheets roll up to form a closed tunnel like structures, something like this, which are called as beta barrels. And these beta barrels are generally found in the outer membrane of bacteria, mitochondria and chloroplast. Now, since these transmembrane proteins, they are in contact with both the extracellular as well as the intracellular side of the cell, so they serve as different functions. Like if we talk about these single pass proteins, they serve the functions of receptors, enzymes, and cell adhesion molecules like adherins, which are involved in cell-cell adhesion. Like for example, here you can see that this is one cell and this is a cadherin protein, and this is another cell and this is the cadherin, another cadherin protein. Now these cadherins are involved in the cell-cell adhesion, or involved in intercellular joining of two cells, right? Now these multipass transmembrane domains, they serve the function of channel proteins, where these alpha helical polypeptide chains, these are very tightly packed to form a closed tunnel like structure, where this exterior side is the hydrophobic and the interior side of these tunnels are hydrophilic in nature, such that they can allow specific ions or hydrophilic molecules to pass across them. And the example of such channels is the aquaporins, which are water channels which allows the movement of water across the membrane. Now, secondly, they also serve the function of transporters. Example, GLUT1 transporter, which is a glucose transporter. Now, these transporters and channels are not the same, they are different. Like, for example, if you see here, for example, consider it as a sodium ion transporter, which has to transport sodium from the extracellular side to the intracellular environment. Now, what will happen? The sodium ion will come and bind to the specific domain of the transporter. And as soon as the ion binds, this transporter undergoes a conformational change and it becomes something like this, and as a result it finally releases the sodium ion to the cytosolic side of the cell, and as it releases it again, this transporter, it again comes back to its original state. So this is how transporter acts for transporting specific molecules across the cell. Now next, these alpha helical multipass domains, they also serve as a function of receptor. Like for example, GPCR, which is G protein coupled receptor, and this receptor is also called as 7-pass protein, where the alpha helical polypeptide chain, it spans 7 times across the lipid bilayer. Next, if we talk about these beta barrels, they mostly serve as the function of channels and transporters. And example of the channels is the purine channels, and transporter example FEP transporter, which is an iron transporter. And these beta barrels, they very rarely serve as function of receptor or enzymes. So this was all about transmembrane proteins. Now let us understand about the other integral proteins, which are lipid anchored and monotopic proteins. So these lipid anchored proteins, these are the proteins which do not insert inside the lipid bilayer, but these are covalently attached to the lipid molecules by a specific anchor. Now, if you look at here, just imagine that this is the extracellular side of the cell and this is the cytosolic side or the intracellular side of the cell. And this is a lipid anchored protein where the protein is attached to the inner leaflet of the lipid molecule via a specific anchor. Now this anchor, it can be either fatty acyl chain, for example myristoyl or palmitoyl group, or it can be either prenyl group, for example farnesyl, right? Now if you look at this another example here, this is another lipid anchored protein which is found in the extracellular side, but this protein is linked to phosphatidyl inositol, which is a phospholipid, by an oligosaccharide chain, right? And this kind of anchor is called as GPI anchor, which is glycosyl phosphatidyl inositol, where this protein is linked to phosphatidyl inositol, or protein is linked to a lipid via oligosaccharide chain. Now this kind of protein is called as the GPI anchored protein and it is found in the extracellular side of the cell. Now you must be wondering that these proteins, lipid anchored proteins, since they are not embedded inside the lipid bilayer, so it is easy to extract them just like peripheral proteins. But the answer is a big no. Why? Because remember, I said that these lipid anchored proteins, these are covalently attached to lipid molecules, and these covalent interactions, as you know, that These are strong interactions as compared to hydrogen bonding or weak ionic interactions, right? So, for extracting these lipid-anchored proteins, we generally use detergents or certain enzymes which can cleave these anchors so that we can easily extract these proteins. So now let's understand the functions performed by these lipid anchored proteins. So they act as enzymes like, for example, alkaline phosphatase, which is a GPI anchored protein found on the extracellular side, and where these alkaline phosphatases, they cleaves or removes phosphate groups from the extracellular molecules. Next, these lipid anchored proteins, they also act as signaling proteins, like for example, RAS or SRC family kinases, which are found in the cytosolic side, where these RAS proteins, they are involved in growth factor signaling in RTK pathways, while these SRC family kinases, they are involved in certain phosphorylation cascades. Next, these lipid anchored proteins, they also perform an important role in cell adhesion and communication. Like, for example, prion proteins, which are GPI anchored proteins. These are found on the surface of neurons where they bind with the extracellular molecules like GAGs, glycosaminoglycans. So, in that way, these proteins stabilize the cell matrix interactions and influence neuron adhesion. Apart from these lipid anchored proteins, they also serve as co-receptors. So this is all about the functions of these lipid anchored proteins. Now let's understand about these monotopic proteins. So these proteins are not as common as transmembrane or lipid anchored proteins, where if you look at here, this is a monotopic protein. Now these proteins, they have alpha helical polypeptide chain which inserts or penetrates in one leaflet of the cell membrane. Now, this region, which is inserted inside, this region is rich in hydrophobic amino acid that interacts with these hydrophobic fatty acid tails, while the region of the protein, which is present outside, this region is rich in hydrophilic amino acids. So, in total, these monotopic proteins are the ones which are partially embedded in the cell membrane. Now, if you understand about these functional role, so basically these proteins, they have this enzymatic role. Like for example, prostaglandin H2 synthase, also called as COX enzyme. So this enzyme, it is a monotopic protein which is found in the membrane of endoplasmic reticulum that is facing towards the cytosolic side. So basically this enzyme, it converts arachidonic acid into prostaglandin H2, and this is a precursor molecule for prostaglandin, which is associated with fever, pain or inflammation in the body. So a common drug which you all might have probably heard of, aspirin, which we generally take during fever or swelling or pain. So basically that drug inhibits this enzyme such that reducing the pain or fever in our body.